Hi everyone. So I'm working on your weekly lecture for quiz 2 this time. And I did get a lot of questions. Oh, I got a lot of questions from you. Some of them I may not get the answers for you, but I will do my best to get the ones that relate to your notes or to your quiz. So what you'll need to know. So first, metabolic reactions. And I want you to understand that a metabolic reaction can be anabolic or catabolic, but it can also be aerobic or anaerobic. And then a lot of them are oxidation reduction reactions. So back to anabolic and catabolic. Someone asked about what are anabolic reactions? What are catabolic reactions? Anabolic reactions are those where you take small parts, let's say amino acids. They're the building blocks of proteins. So you take amino acids, put them together, and build a protein. That would be a metabolic reaction. So it's synthetic. It's synthesizing a protein, something larger, from the smaller units, or the building blocks, the amino acids. All right, so that would be anabolic. How I distinguish these, anabolic and catabolic, is I think about anabolic steroids. Anabolic steroids used for gaining muscle mass in particular. So what are those? Those are synthetic or building up steroids. So anabolic. Catabolic is the opposite. Catabolic would be like taking a protein and digesting it. So you've eaten something, it has proteins in it, you need those amino acids, not the protein from the plant, for example. So your body breaks down that protein into its parts. So catabolic reactions are degradative or tearing down. Aerobic and anaerobic. So aerobic reactions are those that require oxygen. This week we're going to talk specifically about cellular respiration as an aerobic series of reactions. And basically the oxygen is used as a final electron acceptor at the end of cellular respiration. Now that may not mean very much to you right this minute, but hopefully as I continue here it will mean more to you and you'll understand what I mean when I say oxygen is the final electron acceptor. Okay, then anaerobic, just the opposite. They occur in the absence of oxygen. In fact, there are bacteria, for example, where oxygen is toxic to them. It will kill them. So they can only be active when there is no oxygen present. So I'd ask the question, why were the first living organisms anaerobic? Well, it's because there wasn't enough oxygen in the early part of life on Earth to support aerobic organisms. So the first living organisms were anaerobic. Someone asked about cell types. So it's not that easy of an answer. Yes, there are some cells that are aerobic only. They are obligate aerobes, meaning they have to have oxygen for their metabolic processes in the cells to occur. And then there are cells that are anaerobic. They're obligate anaerobes. They can only have their metabolic processes occurring in the absence of oxygen. And then there are cells that can do both. You have cells that can do both, like in your muscles. If you overdo, if you work out beyond your normal level of activity, you can go into what's called an oxygen debt. You cannot get enough oxygen in through your lungs, through your circulatory system, to your cells to do all of the cellular respiration that you need to make all the ATP that you're asking your body for. So they can switch to anaerobic or fermentation. That can happen. And in fact, I ask you for your discussion to look at that video from Crash Course. And even if you don't do the discussion, watch the video, because that is explained there. Oxidation reduction. Someone asked, well, why is something reduced if you add electrons to it? You know, I've been pondering that myself. So I looked it up online, and I'm still confused, so maybe you will be. But it says, because a gain of electrons is reduction, because any loss of electrons by one substance must be accompanied by a gain elsewhere. Yeah, so I'm still, after reading that, I still think it's confusing. So we have to deal with that. That, for example, we're going to talk about reducing NAD+. And it is reduced by adding electrons to it, making it NADH. When we talk about photosynthesis next week, we'll talk about reducing NADP+ to NADPH by adding electrons. I know it's really confusing, but that is just the way it is. These tend to occur together. So one substance loses the electron, one gains it. All right. The one that loses it is oxidized, the one that gains is reduced. There were some questions about energy pathways, and it is important for you to understand substrates or reactants. Let me write that down. Substrates or reactants. So substrates or reactants are what go into the reaction. They're what you start with. And then your end products are what you end with. Okay? So we have this reaction occurring here, this simple one, with a substrate. Then it's catalyzed by an enzyme, and we get an end product. If this was anabolic, we'd have multiple substrates, and we'd end up with a bigger end product. If it was catabolic, we'd be breaking something down, something larger to something smaller. But they're not always this simple. In fact, glycolysis, the Krebs cycle or citric acid cycle, including what I call preparation for the citric acid cycle or Krebs cycle, and electron transport chain are all more complicated. In fact, there are multiple intermediates, for example, in glycolysis. Multiple, it's not just one. So the substrate is glucose. It is acted on by an enzyme, and that changes it to the first intermediate. Then another enzyme acts on it, changing it to the second intermediate. Then another enzyme acts on that, changing it to the third. Do you see where I'm going? It's that these are quite complex. My goal is to simplify these for you, but we'll get to that in a little bit. So intermediates are any compounds that result between the substrates and the end products, and there can be multiple ones. Enzymes. Enzymes are mostly proteins. Enzymes speed up reactions that would otherwise occur by lowering the activation energy, the energy that it takes for the reaction to proceed. So, it is very important that an enzyme has a certain shape because it fits its substrate. So if you think about a car key goes into the lock on the car. That key fits that lock, period. It doesn't fit other locks. It's not made that way. An enzyme fits its particular substrate, and that is based on its shape, just like the keys. If anything happens to change the shape, anything, it doesn't fit anymore. So there are organisms called extreme thermophiles. Extreme because they're in really, really, really extreme environments, in this case, heat. Thermo for heat, phile for loving, so they are extreme heat lovers. They live in thermal vents down in the bottom of oceans. And they are chemosynthetic, some of them, not all of them, but some of them. Some of them are heterotrophic. But they live down there, and basically their enzymes are in their proper shape at a temperature of up to 115 degrees Celsius. Over boiling. If you go down to the bottom of the ocean floor and you grab some of those and start bringing them up to the surface, as the temperature drops, they are going to die because they will not be able to do all their metabolism, because their enzymes that metabolism is dependent on, all their metabolism is dependent on different enzymes. Those can't work anymore because their shapes are changed, so they don't fit their substrates. So it's really, we call that denaturing. You denature a protein, and in this case we're talking mostly about, most enzymes are proteins, when you change its shape. And temperature is one way to do that, pH is another way to do that, so there are others as well. You should be able to recognize something as an enzyme if it has the suffix ASE. So at the end of the word, ASE. In general, that's how enzymes are named. Okay, so they lower the activation energy. Oh, and they're not used up in the reactions. That's pretty cool. All right. Over here, let's look at energy, which is the ability to do work. And when I'm saying work, for this course, I'm talking about all those metabolic processes that occur in a cell. If a cell stops doing what it's supposed to do, most likely it will die. Now, there are some cells that go dormant. Some bacteria, for example, go dormant. In fact, there are some of those obligate anaerobes, remember? Obligate anaerobes, those that cannot tolerate any oxygen. If they're exposed to oxygen, they sporulate and they go dormant so that they can survive that adverse condition of that nasty, for them, oxygen. Okay, now there are a lot of different types of energy, and if you've taken a physics class, or if you will take a physics class, you will learn about other types of energy, like electrical energy, for example. But for these biological processes, we are interested in one type of kinetic energy in particular, and that's thermal energy. So back up a little. Kinetic energy is energy of motion. Thermal energy is heat energy. And basically, the molecules are moving around. That's what makes it kinetic. Okay? Then there's potential energy or stored energy, like chemical potential energy. Now, it's not all chemical potential energy. Electricity can be stored, and that would be potential energy. But we are going to talk about chemical potential energy in ATP. We're going to talk about chemical potential energy in glucose. I'll mention that there's chemical potential energy in NADH and FADH2. So there's chemical potential energy in lipids. There's chemical potential energy in proteins. So just what is this chemical potential energy? And I'm going to abbreviate it CPE. Notice my board is pretty small, and I've got to put a lot on this board to show you cellular respiration. So I'm going to use some abbreviations.